Hi class, welcome to lab. And this lab will be on the cardiovascular system, the blood. And as we talked about in lecture, if we were to centrifuge a sample of your blood down, the plasma would make up about 55%. So after we spin down a sample extremely fast, the plasma would raise to the top. And the plasma is made up of your water, your proteins, your solutes, and then the formed elements, which are your red blood cells, leukocytes and thrombocytes, which make platelets for clotting, will be formed at the bottom. This is a look at your blood cell types. The erythrocytes are your red blood cells, and your thrombocytes go on to form platelets. We're going to focus on your leukocytes. You have 5 types of leukocytes. They will be granulocytes, which are filled with granules or specks, or agranulocytes. And you should be familiar with identifying the 5 different types, especially for your lab exam. So this is what the 5 different types of leukocytes look like. The basophil has this multi-lobed structure. A eosinophil has a bilobe structure. The neutrophil has a multi-lobe structure as well. And these are the 3 different types of granulocytes. And then monocytes and lymphocytes are shown here. The monocytes kind of have a curved nucleus, and then the lymphocytes will be very kind of full in them. Erythrocytes and thrombocytes are the red blood cells and the cells that will go on to create your platelets, but you should be really familiar with knowing the differences between the 5 types of white blood cells and what makes up the characteristics of each. So this takes you through the blood cell and the description. So your erythrocytes, biconcave shape, no nucleus, they'll carry oxygen and carbon dioxide. Your thrombocytes will be fragments of megakaryocytes, and they'll play an important role in blood clot formation. Leukocytes play an important role in defense and immunity. Your basophils, the nucleus will be U-shaped or S-shaped. So this is what a basophil looks like, an S-shaped nucleus. The basophil, the granules will be dark purple depending on the stain and will contain histamine that helps with the inflammatory response. Eosinophils are a bilobed nucleus with the granules staining reddish orange. Eosinophils are elevated during allergies and parasitic infections. So the bilobed nucleus is characteristic of an eosinophil. Neutrophils then, the granules are stained red and blue, usually a multi-lobed nucleus for a neutrophil. And the neutrophil, again, you can see these multi-lobes of the nucleus. They are the most common white blood cell. They make up about 50 to 70% of total leukocytes. The monocytes, so then we get to the agranulocytes. So the monocytes and lymphocytes do not have granules. The monocytes are the largest of the 5 leukocytes. They're almost double the size of the red blood cells. The characteristic of a monocyte is the nucleus is kind of shaped like a kidney or curved like that. So there you have a monocyte. They are elevated in chronic infections like TB or tuberculosis. And then lymphocytes, they're the smallest. They're almost the same size of a red blood cell. The nucleus is round and takes up almost the entire cytoplasm. And they are the second most abundant. And those are the leukocytes. And those are seen here, or the lymphocytes, excuse me. Almost the same type as an erythrocyte, a red blood cell. So spend time in the virtual lab going over the practice for those different lymphocytes, pictures similar to this and from your eLab. And then also part of this lab, you'll have to identify the blood type of different patients with kind of an external link. That's the second part of your Google Lab activity that you'll have to work through. And I will provide you the link on the day of lab. But this is just a review of what your blood type is, what surface antigen will be found on the erythrocytes, and then what antibodies will be found in the plasma portion of the blood. So again, if you're type A blood, you have the antigen A on the surface of your red blood cells, and your plasma will make antibodies against B antigens. If you're type B blood, you have the antigen B on the surface of your erythrocytes, and you will make antibodies against the A antigen. So we call them anti-A antibodies. If your blood type is AB, you have both surface antigens, A and B. So because you have both surface antigens on your red blood cells, you're not going to create any antibodies to try to destroy your own cells. So they have no antibodies. And if you're type O blood, often called the universal donor, because it has no surface antigens and both antibodies in its blood plasma. So again, this was kind of a shorter lab introduction, but you'll spend the majority of your time in the virtual lab um, from Dr. Truong, and you'll also spend time determining the blood type of different patients using the Google Lab activity that I will provide for you at the date and time of your lab. Thanks for listening, guys.